And now I want to talk about resistance. How does this pathogen that was susceptible to the drug, it acted on DNA or whatever it acted on, how does this pathogen become resistant? What is the mechanisms of resistance? So let's talk about that from now. All right, so a couple of vocabulary terms. We can talk about naturally resistant, meaning that the cell just lacks whatever the drug does. It just naturally does this. A lot of times this is where drugs work on bacteria, but they don't work on yeast. The yeast is just naturally resistant. It doesn't have a bacterial cell wall. So therefore, drugs that act on a bacteria cell wall, the yeast is naturally resistant. So that's a type of resistance. Now there's another type of resistance that can be acquired. Say I have a bacteria that has a cell wall. It's been susceptible to a drug that acts on cell walls, but something changes in that bacteria, and now that bacteria becomes resistant to a drug. This is an acquired resistant. The drug didn't cause the bacteria to become resistant, but something happened, a mutation, something happened, and now the bacteria is no longer susceptible. So those are some terms we need to know about. We can talk about resistance and acquired resistance and how that happens, and we can think about historically back when we did our, let me take you back to our lab. Do you remember how we did our Kirby Bauer lab, and we looked at some different antimicrobials and whether they were E. coli or Staph aureus was susceptible or resistant to these drugs. And we found that penicillin, that E. coli and Staph aureus were both resistant to penicillin. And it kind of seems strange because penicillin was our miracle drug. It was that first drug that we had. It was our first defense mechanism. And it's true. Back in the 1940s, when penicillin was first introduced, penicillin killed a lot. Many bacterial pathogens were susceptible to penicillin, and it worked against a lot of things. Now, we've been using it since the 1930s, 1940s. That's a long time. And over time, we have selected inadvertently, but every time we use penicillin, we allowed the ones that were naturally resistant to grow, or the ones that were resistant to grow, and the ones that were susceptible died off. And so nowadays, many pathogens are resistant to antibiotics. And let me show you, I'm going to show you this kind of thing on my low-tech whiteboard to see that happen. So if I consider this being my environment, and this was 1940s, and my black dots are bacteria that were living, black and red dots, this was all some bacteria living in this environment at this time. The black dots were bacteria that were susceptible to penicillin. Before this, we didn't have any penicillin. Everything was just growing. Black dots were replicating. This one red dot I have has just a fluke mutation that made it, that's my black dots, they're replicating and growing. This one red dot has a fluke mutation, and it makes it resistant to penicillin. Now, before penicillin was there, it was no big deal. It was there was no advantage to being resistant to penicillin. So it was living its life. It was just a small variant in the population, and he would replicate just like the others. But then we developed penicillin, which means we came into the environment and we knocked out bacteria that were sensitive to penicillin. Penicillin, every time we used it, we killed bacteria that were susceptible. When it got to these red dots, it couldn't kill it. The red dots were resistant. They had a mutation. They had some sort of change, made it resistant to that. So my black dots went away, but my red dots stayed. Time passed, and bacteria do what bacteria do, which is replicate. These are replicating. My red dots are also replicating, but I still have penicillin. More people are coming in. They're getting diagnosed. I am killing off any bacteria that are susceptible. Can't kill off the red ones. They live. So that continues. Bacteria, time passes. Bacteria replicate. These bacteria also replicate. More antibiotics are used. Patients come in, they're sick. We prescribe penicillin. Kills off anything susceptible to penicillin. Can't kill off the ones that are red. They continue to replicate. Time passes. They replicate. Any of these will also replicate. More time passes, we use more penicillin. It kills the ones that are susceptible. And if you notice, over time, when I look at the percentage in the population, I now have a huge percentage that are resistant to penicillin. So over time, the ones that are left are the ones that can live and reproduce, and they're the ones that are resistant. So the variant in the population, we can change the genetic frequency in the population. The ones that are resistant are now higher are present in a higher genetic frequency from that. So that's what happens over time. It's not that the drug caused this bacteria to mutate. It's that every time we use that drug, the bacteria that weren't susceptible were able to live and reproduce. Okay, so keep that in mind. So this is why we're always trying to develop new drugs and work ahead, because we're constant. Every time we use it, we're selecting for the ones that are resistant. All right, so let's go back to some PowerPoints. This is what we just worked through. How can, in 1940s, all Staph aureus, or about 90%, or nearly all Staph aureus were sensitive, and now they're resistant, and that was how, and that's what we just worked through. All right, so what else do we need to know? Any bacteria that has a mutation, a gene that made it resistant to the bacteria, that will be passed down to all its progeny. So this is vertical evolution, just like when I did my dots and then they live to reproduce, all of the progeny then will have that same gene. This is why I ended up with more red dots than black dots. So this is vertical evolution, and this happens. At first you only have one, maybe a spontaneous mutation happened, but over time, and if we're continually selecting, the only ones that are allowed to live and reproduce are the ones that are resistant, then I have this mutation in all of them. Now remember, bacteria are also capable of horizontal evolution. Remember those plasmids, those conjugation pili, sharing a gene with their friends. So this can make this evolution happen at a much faster frequency. Not only do we have to wait for this to reproduce, which in itself happens very quickly, but this one bacteria that has a plasmid that gives it a resistance to a particular drug, he can share that resistance with his neighbors. And then we end up with other populations that have that and then can pass that along vertically as well as horizontally. So we have to keep that in mind. So resistance can happen pretty quickly. Always keep in mind that this is a selective process. At the beginning, I only had this one that was resistant, but I'm continually selecting for the ones that can live, and that can lead to that change in genetic frequency. All right, so when are we selecting for this? Every time we overuse antibiotics. Every time a patient comes into the clinic and they're sick and they've got a viral infection, but the doctor feels pressured to give an antibiotic, that puts that antibiotic out into the environment. It will select for any bacteria that are resistant to it. That will cause resistance in the environment. Every time people take antibiotics incorrectly, you remember that chart where I said you stopped taking it and it dipped down? That selects for bacteria that are resistant to this drug. Every time we use the drugs in agriculture or livestock and we just throw antibiotics out there, that leads to antibiotic resistance. So this has led to a situation where we have these drug-resistant pathogens left. So we're being very careful now that we understand this. We're being very careful about that and trying to use our antibiotics correctly so that we can keep our weapons for when we really need them for that. This is just another picture with my black and red dots and can kind of show you how that happens as well. So you can look at that as well. Okay, so resistance. We've told you how it happens now. Now we need to look at the mechanisms of resistance. So we're going back into the cell, looking at it at the molecular level. What makes this cell resistant to the antibiotic? So this is back to some cell biology. So don't forget this section. For some reason, I think it's at the end of the chapter. I think students kind of gloss over it. So don't forget this section. Mechanisms of resistance. How can that work? Well, it can happen different ways. We're going to look at some with some pictures. Sometimes it involves enzymes. Sometimes it involves altering the protein or whatever the drug is acting on, the target. Sometimes it involves pumping the drug back out, or keeping the drug from entering in the cell. And then we can talk about biofilm. So let's look at some pictures because I like those better. Okay, so this is a susceptible cell. This is a cell that is susceptible to a drug. So here's my cell. This is the drug, the chemical. I add this to the environment. In this case, the drug goes into the cell easily. The drug has some sort of target in the cell. So this is something the cell does, a protein, some sort of something. The drug binds to this target and then keeps this target from doing whatever it's supposed to do in the cell. And whatever it was supposed to do is necessary to survival. Now that the drug is bound to that, this protein, whatever it is, can't do its job, and the cell dies. So this is what happens to make a cell die. Is it a metabolic pathway? Is it a nucleic acid? Is it part of the cell wall? Whatever it is, the drug binds to it, interferes with it, the target can't do its job, and this cell will die. It is susceptible to this drug. All right, so how does this cell become resistant to this drug? Okay, so something happens. There's a mutation. And when that mutation happens, the drug comes in like normal. We'll do this one first, an alteration in the target. The drug comes in as normal, but this particular cell, the target looks a little different. It can still do its job in the cell is still living, this target still works, but now this drug can't bind the target. So there's a mutation in this target, doesn't interfere with the target doing its job in the cell, but it does interfere with the drug binding. This cell is now resistant. There was an alteration in the target and the drug cannot bind. What else? Sometimes there will be an enzyme that the cell can produce that will inactivate the drug. This is that beta-lactamase. An example of that is beta-lactamase. The cell now has a gene that makes an enzyme, and when that drug comes in, this enzyme alters the drug, and now the drug doesn't work. If a cell can produce this enzyme, that's a defense mechanism. And that can work. Now, beta-lactamase, we said, was an exoenzyme, but still you get the point. It's an enzyme the cell can now produce that inactivates the drug and allows the cell to be resistant. Sometimes there's alterations in the cell, and now the drug that used to come in, the cell has been altered. It has a mutation in one of its proteins or some of its proteins, and now the drug can't get in anymore. So this prevents the cell. This can allow the cell to be resistant to the drug. Sometimes the drug gets in, but there's some modifications, and now the cell can efficiently pump out the drug. All of these are mechanisms of resistance. A spontaneous mutation will happen, means the drug doesn't work as well. That one mutation, remember bacteria replicate in large numbers, spontaneous mutations happen. That one mutation, if it confers a survival advantage, meaning this cell can live when other cells die in the presence of the drug, it will be able to reproduce and then pass that spontaneous mutation on to all of its progenies. Okay, so this is mechanism of resistance. We can talk about that in Zoom if you guys have questions about that. So now be aware that pathogens can require resistance to more than one drug at a time. Remember, we got that horizontal evolution that happens. When we have a healthcare facility and there are lots of pathogens all in the same space, lots of different drugs that are selecting for that, this makes it easier to develop pathogens that are resistant to multiple drugs. If it's resistant to 3 or more drugs, we will technically classify that pathogen as a multiple drug resistant pathogen. Sometimes you'll see them referred to as superbugs, kind of by the layperson. All right, now that we know how this happens, we know that this is an issue, we are trying to protect our patients, trying to do things that make it difficult for these multiple drug-resistant pathogens to happen. Again, our drugs are our defense mechanisms, and if they get around our defenses, then we're back to being killed by bacterial diseases, and we want to protect ourselves from that. All right, so what are things we can do so that we can protect ourselves so that these bacteria, these pathogens don't develop multiple drug resistance? One thing we can do is making sure we take those antibiotics correctly, that we maintain them at the effective concentration for a long period of time. So finish your antibiotics, take them to completion, don't miss doses. those types of things will help us retard the resistance, will help us from creating these drug-resistant pathogens. What else? We talked about synergism. When we talked about using drugs against a metabolic pathway, we described one example that actually uses two drugs. When we can use drugs in combination in that synergism, it makes it hard for, if it becomes resistant to one drug, the other drug can catch it before it has time to reproduce. So we explained that when we talked about that metabolic pathways and synergism. This is a way that we can stop ourselves from creating these multiple drug-resistant pathogens. Only use microbials when necessary. That's why we took them out of antimicrobial soaps for home use. We just don't need them. We don't need to add all those antimicrobials into the environment, into our waterways when it's not necessary. So only use them when they're necessary. You know, hospital settings, they may be necessary. In your house, they're not. They've taken them off the market. And then we're constantly looking at new drugs and discovering new ways of doing that, making variations on our existing drugs to help protect us.